we can see starting from the magnetotactic bacteria, which is shown here, that have these tiny little particles inside the cell, which are made of either magnetite or greigite. And they are organized a kind of a chain inside the bacteria, and they function like the needle of a compass that is helping the bacteria to orient itself along Earth's magnetic field. And then I have these three other examples, which are all marine unicellular algae, and starting with diatoms, that's here in the top. And they make these cell walls out of silica, which is basically glass. And of course, the precipitation of silica is not random, but it has this very sophisticated, very intricate pattern, this nanoporous pattern that if you start zooming in the structure, you're going to see even smaller and smaller pores. And of course, the shape and the general structure of these cell walls is going to be very much like species dependent. The Acantharia is another example that I like very much. And they make these beautiful exoskeletons out of strontium sulfate in the form of senescite. And I find it interesting because the concentration of strontium in seawater is very, very small. And for some reason, it was selected by the Acantharia, and it can selectively filter the strontium from the water and can precipitate it into these structures. And we actually know very little about these because they haven't even domesticated yet, so we cannot grow them in the lab. And it's very hard to keep even the skeletons intact the moment that we take them out of the water. They immediately die, and the skeleton tends to dissolve. And then coccolithophores, which I think at least some of you are familiar with, and they make these scales out of calcium carbonate, where each spoke in one of these scales is one single crystal of calcium carbonate. In this case, it's calcite. And the whole scale is made inside the cell, and once it is ready, it just exocytoses, where it forms this kind of a exoskeleton around that today we still don't know what it is. Mollusk shells are one of the most studied systems in biomineralization. So they also use calcium carbonate to make the shells. It can be in the form of calcite or aragonite, the different crystalline structures of calcium carbonate. We see here how the shell looks like in the scanner microscope. Here at the bottom is the calcite layer, where I have the crystals in the shape of these very long prisms that are hundreds of microns in length, a few tens of microns in diameter. And here at the top, we have aragonite crystals that have the shapes of tablets that are about half a micron in thickness, and they form this continuous layer. And the nacre is basically the iridescent inner layer of the shells that you see here. It's also what herbs are made of. And here at the bottom, another type of another class of mollusks, which are chitons, and they make a living by scraping rocks, searching for food. That's where they get their food. And for this, they have teeth that are made of an outer layer of magnetite, which is the black layer in these teeth here. And it's interesting because we have bacteria using magnetite for navigation. We have chitin using the same material, but for a completely different purpose, which is a mechanical function, which just shows how nature can take the same type of mineral and adapt it and use it for completely different purposes. And that also means that if you zoom in the structure of these teeth, you find something that is very different from the magnetotactic bacteria. It's not just about crystal size. And of course, I have to mention vertebrate bone and teeth, which are made of calcium phosphate in the form of carbonated hydroxyapatite. And again, if we think about vertebrate teeth, which has a mechanical function, grind food, compared to the chitin teeth, same mechanical function, grind tooth, they're made grind food. They're made of completely different materials. And we can understand the reason. I mean, we don't eat rocks, we eat much softer food. So our teeth needs to be made of a material that can fulfill the mechanical requirements of the food that we eat. Maybe if we ate rocks, we'd have teeth made of magnetite or harder stuff. But so it's again showing the adaptability, how we can have same function being performed by completely different materials just based on the mechanical requirements. Now, why are biominerals special, and why is it that we study them? So I like using calcite as an example for this. So if we take calcite and we grow it in the lab, and that's very easy to do. Just take calcium chloride mixed with a source of carbonate, like sodium carbonate or sodium bicarbonate, and we end up with these rhombohedral crystals that after a couple of days are going to be tens of microns in size, which is following very much the crystal structure of calcite. So that's how calcite likes to grow normally. Now, if you look at what organisms do to calcite, I have here an example of a sea urchin spine, which is a single crystal of calcite. It has a very different shape than the non-biogenic calcite, same as the coccolithic prisms that make the mollusk shell. So it's very different shape, very different size. Aspect ratio is something else. And of course, the same with the coccoliths. So that is showing how organisms can manipulate the growth conditions of inorganic materials to completely change the shape from what the crystal structure would normally form according to the crystal structure into something. And it's not just about changing the morphology, it's also about changing the properties. Calcite in itself is a very bad material when it comes to protection because it's very brittle. It cleaves very easily along the 104 plane. So it's not really what you think as a material to use, for example, protection as a mollusk shell does or as a sea urchin spine does to defend the animal, because normally just fracture. But mollusk shell is quite hard. The sea urchin spines are quite hard, quite pointy and sharp, and it can hurt you if you step on them. And what happens is that the organisms cannot just change the shape, but also they can manipulate the material properties and get the material that would normally be very brittle and turn it into something that is much stronger and suitable for the function that it's going to exert. And these are exactly the kind of questions that many people in the field, myself included, were trying to answer. So how does organisms like coccolithophores or mollusks or sea urchins, how do they control the crystallization of materials like calcium carbonate, calcium phosphate, to go from crystals that have one, completely different shapes and two, completely different material properties? And in addition to that, how do the material properties arise from the organization of those crystals into larger structures, which is the case of a mollusk shell, for example, which is basically a polycrystalline material, right? It's an assortment of tiny crystals glued together by organic material, and how that comes together to create the material that you see. So I'm going to discuss the work that I've been doing in these two different systems, the coccolithophores, where I've been looking basically at the mechanisms of coccolith formation, so the formation of calcium carbonate by the cells, and then the brachiopod shells, which I'm looking more into the structure and how that relates to the properties. And I'm going to show why that is interesting. It's actually a very interesting material to study. So let's first talk about coccolithophores. I mean, just for those of you who are not familiar, I mean, I'm personally very interested in coccolithophores. Just looking at the shape of the crystals and that assembly of calcium carbonate crystals, how that happens, I find that mind-blowing. But there's also an ecological or geochemical importance to that, because coccolithophores, they are actually one of the most prolific calcifiers in the oceans. They're the largest geological sink of carbon, and they contribute to the transport of organic matter to the deep sea. I mean, basically, when the cells die, they sink because of the crystals, and they carry organic matter with them that, of course, feeds into the geochemical cycle of carbon. So it's actually quite important. And this is a satellite image of a bloom of coccolithophores. So they're known to form these huge blooms in the oceans. And they can be seen from space, which are all these whitish, light blue whitish patches that you see here. And the reason that we can see that, we're actually looking at the calcium carbonate that is reflecting the light. So we can actually. Another really fascinating fact about coccolithophores is that the morphology of the scales that they make is very much species specific. So we have here 6 examples out of over 100 different species that exist. And we see that each one of them produces calcified scales with completely different morphologies. So that just shows that the control over formation of calcite is genetics. Right? So everything is genetically controlled. Everything happens through specialized proteins, polysaccharides, and templates inside the cell that are going to be determining how the calcite crystals nucleating, how they're going to grow, and how they're going to interlock into shapes. This is a zoom in coccolith from Emiliania Huxley, which is a very well studied species. And again, I have here individual scales and see how they're made of these individual crystals. So each strut in here is one single crystal of calcite. So it means that it nucleated individually and then it grows separately from its neighbor. And we can again compare it to synthetic calcite, and we see that we're talking about a different scale in terms of size. So I have crystals that are a couple of hundreds of nanometers in length in the coccolithophores, and just a few tens of nanometers in thickness compared to a rhombohedral crystal of calcite, which in this case here is 30 microns across. So that again, it's highlighting how they control the crystallization of calcite and how they're changing the shape of the crystals, but also their assembly into these complex structures. These are all questions that I'm trying to answer. So, what we know by now quite a bit about how coccolithophores calcify. We have here in the corner 4 different species. I'm going to be talking mostly about Chrysotila carterae, which was previously known as fluorapatite characterized. We use the old name from time to time, so I can get used to that. And this is a scheme showing a cell. And what we know is that the coccoliths, they are produced in an intracellular compartment that is called the coccolith vesicle. So this is called CV in here. And we know that there are storage compartments that are rich in calcium phosphate. So the calcium, as is taken from the seawater through calcium channels on the membrane, it's somehow translocated into these compartments. We don't know the mechanism. And then from these compartments, it has to be delivered to the coccolith vesicle. Again, we don't know how that happens. And then there are basically 2 types of macromolecules that are assisting in mineral formation here. One is what we call water-insoluble organic scaffolds, so basically a substrate where the crystals nucleate and grow, and we call this the coccolith base plate. We'll talk a bit about this in a minute. And then we have acidic polysaccharides, which are, we call them water-soluble because they dissolve in water. And they're thought to be helping with processes like transport of calcium to the coccolith vesicle, perhaps crystal nucleation, perhaps control over the shape. We don't really know too much exactly what they're doing, but we know this. Now, we also know how crystal nucleation, at least how coccolith formation starts. And the way that it starts, how do we get such intricate pattern, is that we start with the formation of this ring of calcite crystals. So essentially, we have individual crystals of calcite that are nucleating in the form of the ring on top of the base plate, which you don't see. This is a cryo-transmission electron microscopy image. We see the crystals. We see what looks like remnants of the vesicle. So this was a ring that was extracted from the cell and frozen on a thin grid and image. And once you have this ring of crystals, they will start growing preferentially along different directions, which is what the scheme here is pretty much showing until we have the construction. As they grow, they mechanically interlock with each other. Now, I'm going to talk a bit about Chrysotila carterae, which is one of the models that we're studying in my lab at the moment. And they have coccoliths that are slightly different than Emiliania. So this is what happens if you take a whole cell and stick it in the SCM, so it just bursts under vacuum and you get this flat pancake. But that's not an issue because we wanted to image the coccoliths, which we see here more in detail. And we see that we have essentially two types of crystals, what you see in the literature being called the ore unit, or these ones here, they have this kind of a anvil shape. And the reason they're called ore unit is because they have the crystallographic C-axis aligned radially to the direction of the coccolith, so it's basically in this direction. And they alternate with the V unit, which actually has kind of similar shape, but this one is standing up. And the reason it's called the V unit is because the crystallographic C-axis is perpendicular to the coccolith, so it's basically facing up. And here we see how they interlock together. So basically, we have them alternate. Now, I talked before about the coccolith base plate. So that is the substrate where the crystals nucleate and grow. So this is a cryo-TM image of a base plate that was taken by my postdoc. It doesn't look like much. We cannot see much. It's only a 20 nanometer thick substrate, but actually there's quite a lot of structure in there that I'm going to show in a minute. And this is the base plate view with the atomic force microscope. And then you can already see that there is some structure on the surface here. There's some kind of a striation that you see there. And these are TM images. That's work from Mary Marshall, late 90s. And these images basically show the onset of coccolith formation. And what we see is the deposition of calcium-rich particles. That's what we know. We don't know where the carbonate is coming from. It's accumulating around the rim of the base plate. So that's basically around here. And nucleating into calcite and then giving rise to this ring of crystals. Now, one of the biggest questions in the field is how is crystal nucleation controlled? And how do we get crystals with alternating crystallographic orientation? So there's a model that was proposed by Mary Marsh and together with Jeremy Young. And what they propose is that on the rim of the base plate, we have a kind of a macromolecular template, whatever that is, we don't know, that is functionalized with nucleation sites. And they call this a folded ribbon because we have, just think about a folded surface where we have the nucleation sites basically alternating on the two different surfaces. So that's how we would get alternating crystallographic orientations, just coming from nucleation from different directions. Well, we don't know if that is the case. I don't think anyone ever identified or could image this template, but that's what there is at the moment. Even nearly 30 years later, that's what we got. Now, really important piece of work that came, and that was in 2016. It was shown that polysaccharides, so the coccolith polysaccharides, they can direct calcium ions to the nucleation sites on the rim of the base plate. And this is what this AFM image here is showing. So I have basically polysaccharides in the coccolith making complexes with calcium ions and accumulating on the rim of the base plate. And what they proposed at that time was that there was a macromolecular recognition mechanism. So what does that mean? What it means is that for the calcium polysaccharide complexes to accumulate on the rim of the base plate, that means that surface in here must be chemically different from the whatever is in the middle, which makes perfect sense. But also that the binding of the polysaccharides to the surface is specific. So they propose that it's just like a enzyme substrate kind of specificity. So that's where my postdoc Bart came in, because for again going back to the same point, that for the polysaccharides to bind specifically to this region in the base plate, it means that it has to be chemically different than the middle, and we wanted to know what is it that was there. So the first thing that Bart did was cryo-electron tomography. which is a way to image in 3D using the cryo-TM. So basically, he took a base plate from the coccolith, he calcified it, and took the cryo-TM. And using cryo-electron tomography, we got some 3D data sets that, when reconstructed, we could show that the structure of the base plate is actually a lot more complex than it looks like. It has this kind of a three-layered structure where I have this backbone and at the back, here shown in green, which, according to the literature, is probably made of cellulose, but then it's followed by layers of kind of a disordered fibrous material on top, here in yellow, and then another layer on top. So it's like the structure has three different layers. And what is really interesting is that around the rim, we found this kind of a globular structure that is absent from the middle. So there is it's not just chemically different, it's morphologically different, which again, it makes sense, because if we have nucleation sites in here, that means that you must have proteins or polysaccharides, whatever, that are absent from the middle, so that you get nucleation only in this region. Now, another experiment that Bart did was to take, instead of using coccolith polysaccharides, he just took polyacrylic acid, which is mimicking the polysaccharide in terms of the negative charges. So the polysaccharides from the coccolith, they are rich in uronic acid, so they have lots of carboxylate groups, so they're negatively charged. That's how they complex calcium ions. We decided to check whether something very generic like polyacrylic acid would also target calcium ions to the base plate, and it turns out that it does. So this is a cryo-TM image, and all that kind of a fuzzy material that we see around is a complex of polyacrylic acid and calcium ions that is binding to the rim of the base plate. And what this is showing basically is that the binding is not specific. We're using a synthetic polymer here that has no secondary structure. It binds to the base plate with calcium ions the same way that the polysaccharide does. So that means that the binding cannot be specific. So what we propose here is that it's not something specific, but it can be a matter of electrostatic interactions. So all we need is a positively charged surface that can attract another negatively charged complex. And we could confirm that using electrophore dye that is functionalized with this anhydroxysuccinimide group here. And our hypothesis was that if I have positive charges there, then it's most likely that these are positively charged primary amines, and which we can react selectively with a molecule that is functional, a carboxylate that is functionalized with hydroxysuccinimide. And this is actually what we see here. So this is a base plate that was reacted with a fluorescent dye that had this anhydroxysuccinimide, and we got staining around the rim. The stain is not perfect, but it's very much dominant on the rim, and it sometimes is as good as you can get when doing the reaction on a surface. So just to conclude this bit, what we could show here is that the rim of the base plate is functionalized with positive charged amine groups. They provide positive charges that can attract negatively charged polysaccharides. Whether it's the only mechanism responsible for the binding of the polysaccharide, not going back to in vivo, it may not be the only one, but it's certainly, we don't need any specificity. Now, I'll talk a little bit about control over crystal morphology, and that again, using Chrysotila cartera as a model. And the approach that we did here was something slightly different. It was also using cryo-TM and cryo-electron tomography. But just keep in mind the shape of the mature coccoliths, this kind of an anvil shapes for the O unit and unit. So that was the work of PhD student Jessica Walker. And what Jessica did was to extract coccoliths from the cells that weren't formed yet or not 100% mature, and image them in 3D using cryo-electron tomography. This is what you see here, the reconstruction. And that's quite nice because we can see the base plate here at the bottom. It's not completely segmented, but it's enough that we see. We can see fully mature ore crystals fully mature V crystals, we can see how they interlock, which is quite nice. Now, what was lucky for us in that sense is that coccolith formation is not foolproof. Sometimes things can go wrong. And one of the ways that things can go wrong is that we can have a mismatch in the alternation sequence. And when that happens, it's when we have altered crystal morphologies. So this is what we see here, for example. So here I have in purple a V unit, then alternating with an R unit in blue, and then another V unit in purple, in gold. And then we have two R units that are sitting just next to each other. And you see how the shape all of a sudden is very different from what you'd expect from a normal R unit. Right? We don't have this kind of an indentation here in the middle, for example. We just have a kind of a more blocky crystal with sharp interface. And we can also find regions where we have an R unit missing between two V units. We just have three V units next to each other. And what we see is how the crystal morphology changes. Now, the one in the middle, all of a sudden, doesn't have this indentation here in the middle. And where it would be expected to have an indentation here at the back, where this back part here of the R unit would come, it's basically flat. So what this is showing to conclude this part is that we have a number of crystals that are growing in a crowded environment. The V units are going to grow faster along the C-axis, which means vertically. The R units will grow faster along the C-axis, meaning horizontally. And by growing in different directions, different rates, they can limit the growth of the neighboring crystal along different directions. So essentially, we have a system here of basically competition for space that is helping to regulate crystal morphology. The moment that we have a mismatch in the sequence, that competition for space is going to work in a different way, because now we have two neighboring V units competing for space instead of a V and an R unit, and then the crystal morphology is going to change. And to finish the part on the coccolith, I'm going to talk about the work of my current PhD student, Alex Trikas, using ptychographic X-ray computed tomography. This is a synchrotron-based technique that we can image samples objects in 3D using X-rays, but it's different from the way that it works is that you fire the X-ray at a sample, and what you collect is a coherent diffraction pattern. And we just raster the X-ray through the sample, collecting overlapping diffraction patterns. And of course, we do tomography here, so that means that we rotate the sample to scan in 360 degrees all the surface, and collecting those overlapping patterns, which we can then reconstruct to get the 3D object. And this is showing the 3D reconstruction of Emiliania and Chrysotila cartera. So this is basically a slice from the 3D volume where you can see the coccoliths on the outside. That's a forming coccolith here on the inside. And this is a calcium-containing compartment. We see how that reconstructs in 3D. You can have all the coccoliths on the outside, forming coccoliths in pink and some intracellular compartments here. And it's the same thing for Chrysotila. So I have the mature coccoliths in yellow, which or around here. We can see all the organelles, and we can also see forming. Now, but by doing that, what we could get is basically a sequence of showing the different stages of coccolith formation. So we have here, we could divide that into 4 different stages. But ptychography is actually more interesting than that, because what you get, it's not just morphology, but it's a quantitative technique in the sense that every pixel or every voxel in the image is going to have an electron density value, and we can associate that electron density value to the composition of the mineral. And what we can see is that we can actually, based on this electron density, we could divide the coccolith formation into further stages, where I have the first formation of the ring here in red, and then the ring grows in thickness, which is in blue. And then we start looking at the growth towards the inside, and then the formation of the external elements to form the mature coccoliths. Stage 5, it's still intracellular. Stage 6, it's been set extracellular. And we can see that process if you look at electron density maps across the coccolith. Now, what you notice is that the electron density value seems to increase as the coccolith matures. At first, when looking at this data, we thought that could be an indication of changes in mineral, but it turned out that we do have some resolution problems because of the thickness. So we cannot rely too much on the electron density. I mean, if you see electric calcite has an electron density of 0.82 electrons per square angstrom, but you only really get that value when the coccolith is mature and the center. So we cannot really rely on this value, but what we can do and what we're doing at the moment is that we can use the electron density to quantify the amount of calcite that has been deposited at each stage. So that's something that we're working on at the moment, which at least it could give some kind of quantitative picture here of coccolith growth, and plus the sequence, which is quite neat. So now I'm going to move to the work on the brachiopod shells. So the species I've been working with, it's called the Scinisca tenuis. So we see it's here. This is a cross section from the taken with the scanning electron microscope of a polished shell. So you can see here different valves. And it's an interesting system, first of all, because these are shells that are made of calcium phosphate, which it's actually quite unusual for marine invertebrates. Marine invertebrates like sea urchins and mollusks who normally use calcium carbonate to make the shells, and also can also have silica in case of some sponges. But calcium phosphate is really unusual. It's the kind of a material that you find in invertebrates. And but this Scinisca tenuis does use calcium phosphate. It's fluorapatite, so it's fluorine substituted hydroxyapatite, which is actually it's very similar to the bones in our bodies, which is hydroxyapatite, not fluor substituted, but it's still calcium phosphate. The proportion of mineral to organic, it's also similar to what we have. So we have about 65% of mineral, 35% of organic. In the case chitin, proteins, glycosaminoglycans. So similar mineral to vertebrates, similar proportion of organic and inorganic. And that's actually how I started investigating these. I just thought that it's quite unusual and it should be quite interesting to look at, compare it with bone and vertebrate mineralization, basically. But then it turned out that these shells, they have another interesting property, which is that they're quite hard when dry. I mean, they're very thin, so you can break them into quite easily. But for the material, they're a hard material. But if you soak them in water for long enough, and by long I mean about an hour should do it, they become very flexible to the point that you can fold the shell in half and it doesn't break. And this is the image that we see here. That's a shell, an SEM image of a shell that is hydrated and folded in two. And we see that, yeah, it didn't fracture what you'd expect. And we can soak bone in water for as long as we like, and we'll never be able to do this with a piece of bone. We can do that with a calcium carbonate from a piece of seashell or certain spine. And of course, water is part of these materials and contributes to mechanical properties. But we don't have this response to hydration, and we'll never be able to get those mollusk shells and bones and teeth and so on to fold to that extent just by getting them to absorb water. So that means that water is doing something to the structure of the brachiopod shells. It's changing in a way that is leading to changes in the mechanical properties. And this is what I set out to find out. We ended up being a large group of collaborators with this because it turned out a lot more complex than I thought. So we can measure these mechanical properties, so we can actually quantify them. And these are results from nanoindentation, where we have the hardness and the Young's modulus of the dry shell here at the top and the hydrated shell here at the bottom. And we see how both the hardness and the Young's modulus really decrease when the shell absorbs water. So that just again showing now in a quantitative way that the material is becoming much softer. And that is raising the question, what is water doing to the structure? Obviously, it's being absorbed by the shell, but what is it doing? Why are the properties changing that much just because the shell becomes hydrated? So of course, to answer this question, we have to look at the shell structure and have to look at it dry, have to look at it wet. So this is a scanning electron microscopy, the one here on the left, of a dry shell. And you see that at least at the micron scale, we cannot discern much in terms of structure. It's very different from a mollusk that have all those different structural motifs that you can see crystals in the shape of plates or columns and so on. It just looks like a homogeneous structure. And if you look even at higher magnification, it's the same. So it doesn't look very interesting in a way in terms of finding structural motifs there. But if you look at the very high resolution using scanning transmission electron microscope of taking a very thin section out of the shell, we see that the structure is actually more complex than that. I mean, this is a scanning transmission electron microscopy image, and using the high angle annular detector. And what this is telling us is that everything that is bright is inorganic. So that means calcium phosphate crystals, fluorapatite crystals. Everything that is dark, it's not holes in the material. These are organic material that appear dark in this detector. So knowing this, what the image is showing us is that the structure of the shell at the nanoscale is made of crystals or bundles of crystals that are the hundreds of nanometers in length, a few tens of nanometers in thickness, that are surrounded by a network of organic material, which in this case most likely to be chitin. We know that chitin is present in the shell. So that is this at the nanoscale. Now, to find out what water is doing, so we started by doing, again, ptychography. That was a very useful technique in this case. And what we did was to mill a pillar of the shell. Actually, we had to do it at different samples. One we exposed to vacuum, and the other one we exposed to 100% humidity. And what you see here is an electron density map out of a section from the pillar. And here at the bottom, we have the electron density, the histogram showing the electron density distribution in the material. And we see that the value is very much centered around 0.6 electrons per cubic angstrom. And that is right in the middle what we expect from francolite, which would be approximating 0.8, and chitin, which is purely organic, is the main organic component, which is about 0.45. And the reason it is right in the middle is because the resolution of ptychography is limited, and when there are epoxys that have both francolite and chitin, we cannot resolve between them. So we end up with something that is functional of the portion of the two materials in the box. But that's all fine. If you look at the shell that has been hydrated, the electron density distribution is very, very different. We have these bands here now of very low electron density that is now approximating or around the value of chitin, if not lower. And we have now, looking at the distribution, we have now 3 peaks. One still centered at 0.6, but now one at 0.5 and one below. So what this is showing is that the structure of the shell is changing. What water is being absorbed. It's changing at the micron scale. And the most likely thing to be happening is that the organic components are soaking that water and they are swelling. And that's why we see these bands here appearing. And if you look at it in the scanning electron microscope on polished sections, we can see something very similar. So these are secondary electrons that are corresponding backscatter electrons. So everything that is dark, the backscatter electrons image is not a hole, it's basically organic material. And we see that the space between the mineral rods, we can see that here the hydrated one is increasing quite significantly, which means that they're now being slightly further apart, which is again showing the swelling of the organic material. And then we wanted to know what's happening at the molecular level with the shells. And that was done using solid state NMR. And I won't go into all the details on that, but the only thing I want to point out, which is really interesting, is that the difference between the spectra between the hydrated and dry shell is that hydrated one will have the sharpening of the signals corresponding to the N-acetyl methyl and N-acetyl carboxylate groups in the hydrated samples. And what this means is two things. And one is that the chitin chains are becoming more mobile because of water. And the second thing is that we're getting water intercalating between chitin and the mineral, which is acting as a sort of a plasticizer, as a sort of a lubricant, because you're decreasing and making the association between these two weaker. And now all of a sudden, when I apply a load to the shell, we have these two components that can move more freely with respect to each other. So this is a scheme now that basically summarizes. We have the structure of the shell at the nanoscale, which is made of these francolite crystals that are about 10 nanometers in size. They are associated with chitin, chitin, and they're all organized into these rods that are embedded in organic material. When the shell absorbs water, this organic material takes the water, it swells, increases the space between these rods, and also we have water intercalating between the mineral and the chitin, which again is adding the mobility to the individual building blocks. So the whole structure, the different components, they can move with respect to each other when water is absorbed. So that's what water is doing. And I'll just quickly show what we're doing on Lingula. So we're doing similar work on Lingula natina, which is another species of phosphatic brachiopod. It has very similar composition. So shells made of calcium phosphate, 65% mineral, 35% organic. It also has the same response to water, to hydration in the same way. It also becomes more flexible when it absorbs water. But what is interesting here is that the structure is different from the Scanisca tenuis. So we can see in this cross-section in the SEM that the shell has a layered structure alternating inorganic in bright, so the mineral alternating with organic. So what it means is that we have similar composition, similar properties, but different structure. So it'll be interesting to compare what water does to the structure of Lingula and what it does to the structure of the Scanisca. So I'll just quickly show that the shell does absorb water. So I have here two separate experiments where we measured how much the weight is absorbed by the shell just by measuring the increase in weight over time. And we see that the water absorption increases. The shell absorbs about 25 to 35% in weight water. So it does take quite a lot of water. And that takes about 20 minutes for the material to be saturated. And that was a neat experiment that we did. We used micro CT. So we imaged a piece of shell first dry. We did the micro CT. We can have the organic and inorganic layers alternating. So the dark ones are the organic, the bright ones are the inorganic. And then we took the same piece, soaked in water, and then did the micro CT in water. And we can see how the shell swells. And that swelling comes mainly from the swelling of the organic layers. So especially these, what is here shown as band 1 and band 5. So this one and this one, the ones that take most of the water. But the smaller ones are also a little bit limited by the resolution of micro-CT. So at least we know what's happening to the shell at the micron scale, which is that the organic layers are taking the water. And so I have to find out what is happening. So with here, I'm going to finish. So to conclude on coccoliths, to show that I have all those positive charges on the surface of the base plate that are contributing to binding of calcium to nucleation sites together with polysaccharides. We see how how competition for space by crystals growing in a crowded environment is contributing to basically regulating the morphology of the crystals. And we could observe the sequence of coccolith growth and using cryogenic, which is neat. And the brachiopod shows how water changes the structure at different hierarchical levels, and that leads to changes in material properties. And last, I want to thank all my collaborators and present and former group members, funding agencies, and thank you all for your attention.
Thank you very much, Fabio, for this great talk. Now it's question time. You can either raise your hand or directly ask your question by unmuting yourself.
Can I start it off?
Sure.
Mark, I think you have yourself muted again.
Now?
There you go.
Okay.
Yeah.
I knew about coccoliths, but I didn't know about the fine structure there. It's just amazingly how complex they are. My question is, what's the functional significance of that complexity? How different would it be for the coccolithophore to have a structure that didn't have the same overall form to it, but didn't have all that complexity in it? What's the complexity matter?
That's a very good question, and you're touching exactly one of the points that we still don't know, which is the function of these coccoliths, right? And as my postdoc used to say, nature doesn't do things for fun, and the calcification costs energy, so we have to assume that it's good for something. I mean, some species, they can survive without the mineral, without the scale. Some species won't survive if they cannot calcify. We know that much. That's where our knowledge ends. I mean, I tend to think that because the environment in the ocean is obviously not identical everywhere, there are differences in temperature and differences in salinity and chemistry and so on. So I like to think that at least when it comes to evolution, that the environment played a part in the how each species developed its own morphologies, right? That it's somehow related to the environment. More than that, I don't know. I mean, we know that Emiliania has two stages, the haploid and the diploid. The diploid is the one that makes those complex structures. The haploid produces the holococcoliths, which are very different. They're basically calcite, rhombohedral. I don't want to say glued together, but it's not as complex. And so, short answer is that we really don't know.
Well, let me know when you find out, will you?
Yeah, I will.
More questions from the audience?
Yeah, I had a question, if you don't mind. So thanks a lot for the talk. So your idea about the crowding templating, the I think is the original proto-coccolith, or something like that. I think, I mean, to me, it makes complete sense. But then they sort of grow these stalks out, and I don't. And do you have an idea? I mean, it doesn't seem to me to be thermodynamically what should happen to each individual crystal. So do you have an idea for what determines the growth in that specific direction?
Yeah, so the characterization that we did that shows that the crowding templating was on the different species that coccoliths are not as complex. With Emiliania, maybe crowding plays a role, but for sure not the whole story. And there are different hypotheses to that. We know that there are polysaccharides that occlude in the crystal, and they're present more in the extensions than they are in the central ring. So they could be helping to determine that. So that can be one. Another hypothesis that people talk about is that the coccolith vesicle itself may help to template. So basically, you have an external template that already has kind of the shape of the coccolith, and that is limiting how the crystals can grow. And there is another possibility, which could be a matter of calcium gradient. So if you have basically calcium ions being deposited from one specific direction, that is very near to basically a growth front of the crystal, so to speak, you could make the growth more favorable along that direction. This is what we know, and that's that's what I can say about this.
More questions?
Yeah, I have a question. Thank you, Fabio, for this really interesting talk. I have a non-expert question for the first part of your talk. When you mentioned this specificity versus non-specificity of the polysaccharides that transport could you comment on what was the original rationale for proposing this specificity?
So yeah, I mean that was work that was done by another group, and their rationale was that they used the coccolith polysaccharide, and they could get the binding of the formation of this kind of a calcium polysaccharide dense phase on the rim of the base plate, and then they did the same thing using alginate, and it didn't work. And from there, they concluded that it's specific. Now, one of the reasons why we decided to look into it was because, honestly, we didn't buy it, right? I mean, it just sounded like what we're saying macromolecular recognition, which is also the title of the paper, and it didn't really say much, like, just so that's it. Basically.
Okay, thank you.
I have a question that's kind of similar to Mark's question. I don't know if it's known, but you mentioned that calcite, like rhombohedral non-biogenic calcite, is very brittle.
Yeah.
Is there any, do we know at all how the structure of the coccolith, how that changes the structural properties of calcite?
I don't know if anyone measured the mechanical properties of a coccolith. Possibly not. I haven't come across anything like that. I would imagine that it's not as brittle because it has polysaccharides occluded. And that is a common strategy that organisms use to basically reinforce and make the material less brittle. That's basically what makes sea urchin spines, for example, much less brittle than you'd expect, right? Because they act as crack deviation mechanisms. So I would expect that the coccolith is not going to be as brittle as a normal calcite. Now, how efficient it's going to, I don't know. Be interesting to check just for fun.
More questions from the audience?
I have another question. Usually when you grow crystals, you get defects, right? So are these crystals small enough that the probability of defects in each individual, let's say, piece is so small that they're usually individual crystals, or are there defects inside?
They are single crystals. They diffract like single crystals. We've done nano-diffraction on basically on the struts that they're poking out, and it is a single crystal. Now, I don't know what is the coherence length in there, so I don't know the defects that they have in there. That I don't know. There is a paper where they did high resolution powder XRD on the coccoliths. They do look at lattice strain, but they don't say much about coherence length in there. And I know that we've done that as well on Jafra capsule oceanica, but I don't remember. They do have defects in there, but I don't think it could be more related to the organic. I mean, in the end, it's difficult to know if the defect is coming from the organic or if it's coming from the crystal itself. That's one of the issues there.
Okay, thanks.
More questions from the audience? Okay, there seems to be no more questions. So I think we can thank again Fabio for the great talk. We're approaching 6 p.m. and